You guys are so easily entertained. It's unbelievable. Anyway, that was a creation of Anna, Erica, and I don't know who, but a lot of students and technical people, and it was a lot of fun, I take it, and at low cost. Yes. So welcome to CBR Research Day. This is the fourth time we've done it in this manner, but it's the fifth time we've actually had a CBR Research Day. We started this off with about 4 or 5 students supported by various organizations, and then it was really a sort of a popular, gained popularity, and actually we really appreciated the value. So we've expanded now. We support over 25 summer students, and these summer students are supported primarily through the generous donations of our sponsors, in particular Pfizer, CSL Behring, Grifols, Biogen, Canadian Blood Services, Bayer, Macropharma, Alexion, the Sheldon Naiman and Linda Vickers Hematology Endowment Fund, and the Neil Mackenzie Memorial Fund. Over the years, we've had other donors, and we welcome more. These donors provide salary support for these summer students, at least partly, and then the rest comes from matching funds from the supervisor. The summer students are supervised not only by PIs, actually probably rarely by PIs, but more often by technicians, research associates, postdoc fellows, and actually we've integrated that into the program to formally give these people opportunities to mentor, which is really, a great opportunity that we don't give our students enough of. So it's been a great program. So the day goes as follows. The students give 3-minute presentations, so these are pretty snappy presentations. They're allowed 5 slides each, no animations. I see a couple that have snuck in a 6th slide, but it's really 3 minutes, and I will yank you off the stage, and you will hear a noise if you're not on time, so please be prompt. All right, so as they're making the presentations, the next one will come up while the wild applause is going on for the previous one. In addition, after the 3-minute presentations, of which there are 28, but it will fly by, I promise. We have the Neil Mackenzie Award. It will be a mentorship award, which will be presented by Dieter Brumme. There were, and you'll hear a little more about this award, there were over 20 nominees by over 30 nominators. So it's you'll hear a lot about it. Many of you knew Neil. This will be followed by a keynote speaker, and Ross will introduce Bill Milsom. So we have a great day. At the end of this formal part of the day, we'll move into the atrium where there's all kinds of posters being presented, not just by the summer students. These will be scored by postdocs and research associates. Just as a reminder, the students will score themselves today. All right, so encourage the students to score themselves very highly, okay? And there will be awards given based on oral presentations, and then later on this afternoon on poster presentations. So in addition to the posters, of course, there'll be a pool set up for everybody to go swimming, and lots of alcohol and non-alcoholic beverages for you to enjoy yourself. There'll be a barbecue at the end of the day. So we are going to start. Now, I'm going to ask Claire to come up. Claire, okay. You all have the order, all right? So as soon as Claire is finished, the next speaker will come up during the applause, so we don't spend too much transition time. One sec. We ask the speakers to stand exactly where I am, essentially, okay? Use the mic, because this is being webcast all over the world, I know for sure. I know for sure that there are people watching in Scotland, hello, and other places in the world. All right, so please speak into the microphone and use the key of the computer forward and backward, hopefully only forward, okay? And also, if you want to use the pointer on the keypad, all right? Got it? Yeah. Okay, Claire, you're off.
Great, thank you. Hi, everyone. My name is Claire Wu.
Real loud.
Okay. This summer, I had the privilege of working with 3 hematopathologists across 3 hospital sites. I worked at BC Children's Hospital, Royal Columbian Hospital, and at Vancouver General Hospital. The title of our project is a multi-center audit of non-surgical red cell transfusions in adult emergency department and neonatal intensive care unit patients. Briefly, red blood cells are transfused to increase oxygen delivery to the tissues, and so they are a mainstay of treatment for anemias. However, these transfusions are also in short supply, they are in high demand, and they are also quite costly. And so we were interested in determining how often physicians were transfusing appropriately. And so the purpose of our project was to create and to use an online audit tool to collect clinical information from the recipients of these transfusions, and then to determine if these transfusions were appropriate or not. In terms of our methods, we took all RBC transfusions in the calendar year 2014. We then took out all surgical transfusions, and then based on the non-surgical transfusions, we determined the target sample size. We then took the patient list and collected clinical information using the electronic medical record and the paper charts, and we put that information into a secure web-based tool called REDCap. The appropriateness of these transfusions was then determined by one of 4 hematopathologists. So we have only some preliminary results to share with you. We were able to audit 193 NICU charts, of which 52 have been looked at by a physician. The point of these pie graphs is to show you the groups of information that we were able to collect, including the most responsible diagnosis, signs and symptoms at the time of transfusion, comorbidities, and things like that. The table on the left side here shows you the results from those 52. 36 were deemed to be appropriate, while 16 may require further review. We were also able to collect some demographic data, such as the age and the weight of the patients, as well as some lab results, such as the hemoglobin levels. And so our next steps are to continue to do audits on the NICU and the adult populations to establish the baseline rate of appropriate transfusions, and to hopefully identify suboptimal transfusion practices that can be targeted with further education. Thank you.
Good afternoon. My name is Andrew and I am from Dr. Brom's lab. I will be presenting the selective inhibition of the collagenase and elastase activity of cathepsin K with tensionodes. The protease cathepsin K, shown here, is critical in the degradation of the extracellular matrix. It is a possible drug target for the treatment of osteoporosis and arthritis. We screened 31 tensionodes, which are low molecular weight compounds extracted from the plant Salvia malterhiza used in traditional Chinese medicine. 4 of these tensionodes are shown on the right. First, we confirmed that the tensionodes bind to the ectosite of the enzyme rather than its active site. The goal is to selectively inhibit the collagenase function of the enzyme while leaving the other functions intact. We added a substrate that we know is cleaved by the enzyme's active site. In a large excess of the inhibitor to the enzyme, there was at most a 35% inhibition of the active site's activity by some tensionodes. Next, we performed an elastin degradation assay. We used elastin Congo red, which when degraded by the enzyme, releases a red color. We measured the absorbance, and compared to the controls, the bars in red indicate at most 25% degradation. In other words, there was more than 75% inhibition. We ran similar assays for soluble and insoluble collagen. Again, the bars in red indicate the tensionodes that inhibited the collagenase activity the most. For the insoluble collagen, we measured the degradation using hydroxyproline analysis. Hydroxyproline is a post-translational modification of collagen, and it directly reflects the amount of collagen present in the supernatant. We viewed the insoluble collagen fibers closer using a scanning electron microscope. In the top left is an untreated control without cathepsin K, and the two pictures to the right of those show complete degradation. There were no inhibitors in those. The remaining pictures show varying degrees of degradation in the presence of the tensiones. We determined that there were some tensiones that were potent inhibitors of the elastase activity, but not of the collagenase activity, such as T14, 19, 24, and 26. We also found tensiones that did the reverse, such as T17 and T20. We also found tensiones that inhibited both these activities. From this, we can conclude that the tensiones must bind to the enzyme at different sites to account for these selectivity differences. Thank you.
Hi. My name is Luca, and I'm a summer student in the laboratory of Dr. Dana Devine. Here today, I'm going to present the summer project that I've been working on that I've titled Safe Walk for mRNA. Platelet mRNA is transported to microvesicles via stress granules. First, I'd like to give you a background about the theory behind what I've been doing. So if you look at figure 1, you can see what we know about platelet microvesicles so far. You have a platelet, and upon activation, the platelet will release small microvesicles. These microvesicles contain RNA and they contain protein. The exact composition of these RNA and proteins is not entirely understood. Figure 2 describes what we're trying to figure out. The aims of the study are a few things. First, we have platelets. We want to discover if these platelets contain stress granules. Stress granules are small, dynamic bodies that are present within cytoplasm that are formed upon cellular stress. Next, we want to see if these stress granules are transported to these microvesicles upon platelet activation and stress. So the methodology of this is fairly simple. You start simply with platelets, and you give them an agonist. In my case, it was an apoptotic agonist. This produces microvesicles, which you can lyse, extract mRNA, perform RT-PCR, and get, hopefully, a really pretty agarose gel. However, as most summer students would understand, starting in a new lab requires some optimization, and this comes with hurdles. However, in my case, these are more like mountains. So a significant part of my summer, as this is a presentation of my summer project, was spent overcoming these mountains, finally getting to the top of one, only to see there's another in your way. And you finally get to the top of the next, and there's your perfect assay sitting on top of the tallest one. So some preliminary results that I can share with you are some mRNAs that were found within platelets. This is the results of an agarose gel that was conducted on cDNA extracted from platelet microvesicles. As you can see, there are some various transcripts that I've identified, such as PF4 and GAPDH. However, if you'd like to know more, I'd like to ask that you come see my poster. Safe walk is not really required.
All right. Awesome. Hi everyone, my name is Tony Fang and I'm a summer student from the Devine Lab. So what if I told you there's a new fast way to die? Yes, to die. Anyone interested in dying? Any takers? No? Oh, okay. Whoa, okay. We don't want to make the headlines death by oral presentation, now do we? Not good. But don't worry, the death I speak of is in platelets. And lucky for you today, I will be your wonderful guide to this lovely tour. Let's start with the basics. So it's already well established that in mammalian cells, they can undergo the extrinsic pathway of apoptosis via the Fas receptor. But nobody had ever investigated the idea of a fully functional Fas receptor in platelets before. Interesting, eh? So here's a glimpse of what I mean. Here you can see a validation of the Fas receptor in platelets using flow cytometry and also by using immunoprecipitation and Western blotting. So now that we know the Fas receptor is present in platelets, that's awesome. But as scientists, our jobs are never really done, are they? Unless you get fired. But that's another story. So to take that idea further, we're going to take it back to the mammalian cell and use that as a model. So you can see here the FAST receptor can be activated by an artificial agonist, namely CH11, or the more physiological FAST ligand. And upon activation, there's a downstream cascade of the cell signaling pathway, most notably the cysteine proteases caspase 3, 8, and 9. But keep in mind, this is a model, and the same may not be true in terms of platelets. So here's one component of our study design, where we've added the fast receptor inhibitor ZB4, a fast receptor agonist CH11, and also both. Simple enough. But what does that lead up to then? Well, we know the fast receptors in platelets. Awesome. But if you'd like to know the results of the following experiments, you have to pay $60 for the full tour. Yes, $60. What can you expect in the full tour? Well, you can hope to learn more about the relationship between RNA and the fast receptor in platelets. You can hope to learn more about the time-dependent cell signaling pathway. You can hope to learn more also about how ADP may indirectly trigger apoptosis and much more. But, since you've all been such a great audience today, I will give you the limited time CBR discount. A free tour to my poster. Yes, free. Calm down, it's free. Thank you very much, and up to semester.
Good afternoon. I'm also from the Divine Lab, and my name is Wallace. So I'll be telling you a bit about a problem that the Canadian Blood Services has been having and the preliminary steps that I've taken to solve this. So, in a quality measure of platelet concentrates in the CBS inventory is looking at pH. So over the years, they've noticed that there are several apheresis platelet concentrates in their inventory that repeatedly fail quality control, and that's to say that they have really low pH. And they've also identified a of donors who repeatedly donate these low pH units. So our goal is to assess whether or not there are any donor-specific differences in these platelets that might allow us to identify whether or not they'll be donating poor platelets. So throughout the summer, I've been looking at normal platelets and I've been assessing degranulation, and that's an indicator of platelet activation. I've also been looking at phosphatidylserine flipping, and that's an indicator of apoptosis. I've also assessed pH and lactate. And then we hit a brick wall. As we all know, brick walls aren't there to stop us, they're there to stop those who don't want the answer bad enough. So what have we done? Well, we thought about whether or not there are any other donor-specific differences that we could assess, and whether or not there might be a metabolic basis to all this. So we focused our attention on glycolysis, and in particular, we looked at anaerobic respiration. And the reason behind this is we know that pH and lactate are highly correlated. So whenever there's a high lactate concentration, pH usually is very low. And so we conducted a lot of glycolytic protein Western blots. We also decided to inhibit the reduction of pyruvate into lactate in the hopes that we could assess how much of pH is actually attributed to lactate concentration. So we found this salt called sodium oxamate, and it's an analog of pyruvate. And I basically took a pH unit bag and I split them into two smaller mini bags. So one of them I had the inhibitor and the other was a vehicle control. And what we found was actually quite astonishing and certainly surprising. We actually found that in the inhibited treatment, pH fell faster than in the control. So now if you'd like to find out more about why this might be, please visit me at my poster afterwards. And I'd like to take this time to thank the CBS, CBR, and the Divine Group for all that they've done this summer. Thank you.
Hi everyone, my name is Cody and I'm with the Carson Lab. My project is the AML 2MP database. So let's start with what is AML. AML, or acute myeloid leukemia, is a cancer that starts in the bone marrow. It affects cells that are supposed to turn into different kinds of blood cells. With the AML, the blood cells turn into cancer cells that start in the bone marrow but can quickly spread into blood. And if not treated, it can be fatal within a few months. And this is where the PMP, or personalized medicine, comes in. With the PMP, we're aiming to provide an efficient treatment upon diagnosis of by examining the tumor DNA. As you can imagine, that to enable this, we need to store a lot of information about these specimens and in patients. With so right now, the database contains more than 20,000 samples, the specimens. And these specimens and information come from different data sources in different projects and many times in different formats, which means it's very difficult for researchers to actually retrieve this information. And sometimes it's even hard to find this information. With the ML-PMP database, we're aiming to provide a uniform way of retrieving, validating, and storing this information. And to do this, we developing tools for the importing and the different ways to use the database. And we not only do we store information, but we also store sources where the information comes from. And this means that we can automate the process of validating this information to ensure the correctness of the data. So right now this is the structure of the database. We are storing patient information, specimen information, and the library analysis information, and many, many more. With the hopefully with the ML-PMP database, we can pave the way for further PMP research. And thank you.
Hi there. My name is Paul, and I'm from the Hancock Lab. So today I'm going to be talking to you guys about D-peptides as novel antimicrobial and antibiofilm agents against drug-resistant bacteria. Now, to start off, to put things into perspective, if we continue on the pathway that we are right now, drug-resistant infections will kill approximately 10 million people per year by 2050. It's a pretty high number, yeah. So this is why this research is quite important. In the context of human infections, we see that biofilms are actually associated with approximately 2/3 of them. And because of this, it's very important that we have drugs that target biofilms. But unfortunately on the market today, we actually don't have any. So in the lab, we study antimicrobial and antibiofilm peptides. And specifically this summer, we were studying D-peptides, D-amino acids. And these peptides exhibit potent broad-spectrum activity. They also are protease resistant, meaning that they have a longer half-life in the human body. So this makes them quite useful for use as systemic treatment potentially in the future. So one of our lead D-peptides called DJK5, we found that DJK5 kills faster than clinically relevant antibiotics. Now we took DJK5 and we took 4 other antibiotics and we tested them against MRSA, or methicillin-resistant Staphylococcus aureus. And we found that DJK5 actually kills much faster than all 4 of these antibiotics. And as you can see by this graph, it's quite a substantial amount too, which is great. It provides a proof of concept that potentially peptides can be used in the future as systemic treatment. In addition to which, we've also found that DJK5 inhibits and eradicates biofilms. So the first picture that you see on the left is a MRSA biofilm that's grown for 3 days. And the second picture is what happens when you introduce 2.5 micrograms per milliliter of DJK5 to the minimal media that we use to grow the biofilm. And you see that there's complete inhibition of the biofilm. And in the last picture, what you end up seeing is we grow a biofilm for 2 days initially, and then we introduce DJK5 after the second day. And the same concentration eradicates the 2-day biofilm that's formed. So it's quite exciting. It's also very interesting in terms of clinical relevance. So we're also starting to do synergy experiments with other antibiotics in DJK5, and we found that you can lower these concentrations even lower and then get the same kind of inhibition and eradication. So it's very, very exciting work, and we're excited to see where this goes. If you have any questions, please come by and talk to me on my poster. Thank you.
Hello, everyone. So my name is Raisa, and I worked with, I didn't have this font initially. Okay, so I worked with Dr. Jeffries this summer, and I study immunology, so I will give you some background right now. So anytime there's some sort of a viral infection or tumor in your body, the only way you can get rid of it usually is with something called the CD8 response. And this begins with something known as MHC class 1 that is presented on our dendritic cells. Now, dendritic cells, they're a very rare subpopulation of cells, but they are arguably one of the most important in our body, because if we didn't have this, then there would be no way for our body to activate the specific immune response. So in the context of viral infections, MHC class 1, what it does is it actually activates these CD8 cells, and then the cells actually target the infected cells. And so there are two ways MHC class 1 can actually present these peptides to to the CD8 cell. And the first way is the classical pathway, which is where, you know, the cell itself is infected and the normal cytosolic protein is actually presented on the MHC class 1. But sometimes, you know, it's a very crucial thing that happens. It's called cross-presentation. And in this situation, what happens is, because dendritic cells are so rare, the possibility of the dendritic cell itself getting infected by a virus is actually quite low physiologically. So what happens is that it phagocytoses a dead cell. And normally it would present phagocytosed peptides on the MHC class 1, but it actually shuttles it, as you can see over here, using the MHC CD74 complex, and brings it to the endolysosome where the phagocytosed necrotic cell meets it. And over here, this exogenous peptide is actually presented on MHC class 1. So with this in mind, what I sought to study was, is there a way that I can potentially take this trigger for cross-presentation and use it for vaccines? Because there are a lot of antigens that may be potentially immunogenic, but we don't really know because we can't really present it enough to study them. So the first thing that we did was the first step was actually to do a proof of concept testing. So to see that CD74, we're actually going the right path, we conjugated it with something that's very well characterized as an antigen, and it's called OVA. And we took the transmembrane domain of the CD74 right here, and it's also called invariant chain. And we, because it contains an endosomal targeting signal, which as you saw is key to the cross-presentation, we conjugated that with ovalbumin, and we compared how the MHC surface level expression is in cells that contain CD74 compared to cells that don't have CD74. So this is how I generated the vector, and because my end goal is to sort of look at how this affects vaccines, I paid close attention to considering adenovirus, because this is actually a very safe option that's already been used as gene therapy, as well as vaccines for influenza as well. So, you know, it's just basic transformation of E. coli and then passaging it through a certain set of hex cells, which contain the other missing component to actually create fully infectious viruses. And then after that, I infected dendritic cells. And interestingly, what I saw was actually quite fascinating, in my opinion, and that is that the levels of CD74, the levels of MHC class 1 in OVA CD74 dendritic cells was actually far higher when compared to OVA infected dendritic cells only. So if you look at, so these are the double positive quadrants. So over here, this is showing us the MHC class 1 levels. So 0.81 for OVA CD74 and 0.32 at the same time point with the same number of viruses for just OVA alone. So this is just one of the preliminary results, but we found similar patterns for different portions of OVA, whole OVA, as well as just the synfecal peptide. And what I hope to do is to actually move on to clinically relevant clinically relevant antigens, such as the influenza M1, which is a universal antigen, meaning that if you can find a way to create the perfect cocktail of M1, actually eliciting a proper response in your body, you could potentially need to only take one flu vaccine your whole life for any strain. So that is actually what I have spent most of the summer working on, lots of troubleshooting, as other people have explained. Also, I had spent 2 months working with a defective antibody, but I got refunded, so that's good. But yeah, that's pretty much it for me for now. I would like to continue to actually work with an in vivo model. One of the main reasons I actually chose influenza, aside from it being very prevalent clinically, is because we do have a very well-established mouse model. So, you know, for my director studies coming up this September, I feel like I am very well postulated to make some really good progress. So thank you, CBR, for this opportunity.
Commercial break. Have to change the slides. You can come on up.
My name is Arshdeep, and the title for my project is Tangential Flow Filtration Protocol for HPG, Purification for Peritoneal Dialysis. So I'll start off by explaining what peritoneal dialysis is. And basically it's an alternative to hemodialysis when kidney transplantation isn't possible. So what happens is that a catheter, which is tubing, is attached to the patient's abdomen. And through that you fill the abdominal cavity with a solution of high osmolarity. And then basically the waste from the patient is absorbed into the solution through the peritoneal membrane. And then you drain the solution through the catheter. And the most common osmotic agent in PD solutions is glucose, but that has adverse side effects such as obesity, hyperglycemia, and the others listed above. So basically an alternative to glucose would be low molecular weight hyperbranch polyglycerol, which is pretty easy to synthesize as shown above. But it also has drawbacks in that the purification is tedious, expensive, and time consuming, and it's not scalable. So the aim of my project was to develop a tangential flow filtration protocol as an alternative to dialysis tubing for the purification of HPG. So above you see the actual apparatus that I used, and below is a schematic. So basically you load the HPG right here, and the pump pushes it over a membrane, and the filtrate comes out here, and the rest of the solution circulates back through the system. So the methodology of the experiment was to vary 3 variables and try to optimize the conditions. And basically we varied the pump speed and the transmembrane pressure, which is the pressure on the membrane by the use of the clamp. And we also varied the actual concentration of the starting material itself. So here's some results, and basically the graph on the left shows the pump speed plotted against the percent loss. And so the blue line is after 1 cycle, and the red line is after 2 cycles. So basically from the first graph you see that around pump speed 20 you get the least amount of polymer lost. And beside is a graph of conductance, which can be used as an indication for the amount of ionic impurities in the polymer. So basically that graph, the conclusion you can get from that graph is that 2 cycles are needed to get to dialysis tubing levels of purification of the polymer. And over here you have the variance of the transmembrane pressure, which shows that as the transmembrane pressure is increased, you get a slightly higher yield. The highest one that we could have done was 27.5 psi. And on the graph on the right shows the variation in concentration of the initial starting unpurified polymer. And it shows that as you decrease the concentration, you get a higher yield. So the optimal conditions I found in summary were 20 RPM for the pump speed, and transmembrane pressure at 27.5 psi, and the initial feed concentration of 1.24% by weight in water. And the gel permeation chromatography data showed that after one cycle, you achieve the desired molecular weight of 1 kilodalton. And also, since the conductance needs more than one cycle to reach dialysis levels, you have to trade off the yield if you go past one cycle, so it's recommended that some other step be required to get rid of ionic impurities in the future. And other future directions, I would recommend the use of a smaller membrane with smaller pores to get a higher yield. And also running the filtrates through the system again might improve the yield as well. And also the further optimization of this protocol should be pursued because it is scalable, it's more cost efficient, and it also has potential for application to other soluble macromolecules that need to be purified. So thank you.
Just a reminder that you have a pointer here, okay? Just a reminder for the speakers that you guys have a pointer on the touchpad, okay? So it's just easier, okay? That was great.
Hello everyone, my name is Braden Wilkinson, and I'm going to be talking a bit about the research that I've been doing this summer in the Castrop lab. And my research is based on the ongoing research of my mentor, Stephanie Nowakowski. Her work focuses on utilizing nanoliposomes as delivery vectors for nucleic acid-based treatments of cardiovascular diseases. Now, we already know that, we've already shown that we can deliver RNA to platelets through nanoliposomes. However, we've yet to determine whether or not what we're delivering is full-length functional RNA through this mechanism of delivering RNA to platelets. As well, we've yet to determine the site of RNA localization post-delivery. As such, this requires a mechanism through which we can visualize and tag the RNA that we deliver to platelets through this mechanism. As such, we proposed utilizing RNA containing the spinach 2 sequence shown here. This sequence in particular has been shown to form an RNA aptamer that interacts with the small molecule DFHBIT. And basically, essentially what this does is it shifts the fluorescence of DFHB1T to a range that's more, that resembles GFP. Now, the objectives are as follows. Our objectives were to synthesize a luciferase spinach 2 plasmid construct, to encapsulate RNA that we transcribed from that construct in nanoliposomes, and to subsequently deliver the encapsulated RNA to platelets and visualize the spinach 2 induced fluorescence. Our hypothesis was that luciferase spinach 2 RNA, once paired with DFHB1T, would be detectable when delivered to platelets. To synthesize this construct, we inserted the spinach 2 sequence in between varying pairs of restriction sites in a luciferase-containing plasmid. This process yielded 2 different plasmid constructs, a luciferase spinach 2 plasmid construct and a spinach 2 plasmid construct. We then proceeded to individually assess the fluorescent and luminescent capabilities of the spinach 2 and luciferase sequences found in our construct RNA respectively. Unfortunately, the relative fluorescence of the DFHB1T fluorophore incubated with our spinach 2 luciferase construct was significant, or unfortunately not statistically significant in its fluorescence. However, we then proceeded to perform a cell-free expression assay that concluded that the luciferase produced from the luciferase spinach 2 RNA was significantly luminescent. From there, we hypothesized that an identical mutation in both of the spinach 2 sequences found in our 2 constructs were essentially inhibiting the formation of the RNA aptamer commonly associated with spinach 2. As such, we then proceeded to perform site-directed immunogenesis to remove this sequence. Through that, we were able to correct a single mismatch in the spinach 2 construct. Unfortunately, we're still working on deleting the identical nucleotide in both of the constructs. However, once we achieve this, we will then proceed to reevaluate the fluorescent capabilities of our spinach 2 construct. Thank you.
Okay, so hi everyone, my name is Calvin and I work at the Chem Lab, and today I'm going to present you some of the work I've done this summer relating to periodontal disease. So periodontal disease, such as gingivitis, is one of the most common dental problems that adults face as they age, and it comes with a variety of complications, as you can see above. So at the cellular level, periodontal disease it develops due to the accumulation of bacterial plaque over time, which causes host cells, such as human gingival fibroblast cells, to secrete destructive enzymes such as MMP1. So what our lab is trying to figure out is how do these host cells communicate with each other biochemically. So what we do know, thanks to Dr. Javed, not Ahmed, I spelled it wrong, is that a major platelet cytokine called platelet factor 4, when treated to human gingival fibroblast cells, actually results in an increased expression and secretion of MMP1. And this was confirmed through both ELISA and qPCR. So the aim of my work and the role I play in this project is to try to figure out what's the internal signaling mechanism used by human gingival fibroblast cells to respond to platelet factor 4 treatment. And to accomplish this, I needed to analyze several different internal signaling pathways. So essentially to do this, I had to do a lot of Western blots. So after culturing human gingival fibroblast cells and treating them with and without platelet factor 4, thanks to Dr. Javed again, we lysed them using detergent and then detected key members of the pathway through Western blotting. So through several rounds of Western blots, I was able to get this result. So if you focus your attention, or actually to start off with, for most of the proteins of each pathway, the relative protein level did not change after platelet factor 4 treatment. But if you focus your attention to the right, specifically to the phospho-ERK1-2 pathway, and in particular the phospho-P4442 MAPK, you can see that there's actually a sharp increase in the protein level after platelet factor 4 treatment. And we knew that this experiment was successfully done since our loading control GAP-DH was evenly loaded. So what we can conclude from this result is that phospho-P4442 MAPK, which is a mitogen activated protein kinase, is upregulated in response to platelet factor 4 treatment. And this makes sense to us because downstream targets of this kinase are transcription factors, which may subsequently be the reason why we observed an increase in expression and secretion of MMP1. So the next step of our study is to try to figure out which which receptor binds to platelet factor 4. So yeah, that's it for my talk, and thanks for listening.
Hello everybody, my name is Cameron Perro and I'm going to tell you about the work I've done in Dr. Ed Conway's lab over the past few months. So age-related macular degeneration is a debilitating disease of the eye and is the leading cause of blindness in adults over 50 in the Western world. It is caused by excess complement activation and oxidative stress, which lead to chronic inflammation. This damages the choroidal endothelium and the retinal pigment epithelium, the two cell layers sitting just beneath the retinal photoreceptors, and leading to the dramatic vision loss you can see on the side. Current treatments are invasive and costly. However, polyphosphate, a linear inorganic polymer, was recently found to suppress the complement cascade. And work done in our lab has actually shown that poly-P serves to relieve some of the stress associated with excess complement in an in vivo model of AMD. So the main goal of my project was to characterize the effects of poly-P on the oxidative stress associated with AMD. Specifically, we wanted to understand how oxidative stress affects the cell junctions of these cells, as the damage to these cell junctions plays a vital role in the pathophysiology of this disease. So what we did was we first treated these cells with oxidative stress plus or minus poly-P, and then stained the cells for the cadherin cell junction protein and used confocal microscopy to image this. As you can see, we gathered important qualitative data, but it was highly variable, so we couldn't draw any conclusions. To get more decisive conclusions, we moved this experiment, a similar experiment, but rather than using confocal microscopy, we used a Celomics array scan. And what we did was a similar thing where we treated the choroidal endothelial cells with peroxide plus or minus varying concentrations of poly-P, and then the Celomics array scan quantified the fluorescence of the cadherin staining. As you can see on the right side of the graph, Poly-P provided a dose-dependent protection of the cadherins in under 100 micromolar concentration of peroxide stress, and it also seemed to actually upregulate the expression of the cadherins junctions without oxidative stress. This finding led us to question whether polyphosphate might actually induce endothelial cell proliferation, as this would be a hazard in treating poly-P because it could further exacerbate the chronic inflammation. Using a BRDU cell proliferation assay, however, we found that 3 different levels of poly-P did not significantly induce or enhance cell proliferation in an endothelial cell line. Together, the protection of the cadherins junction by poly-P and the lack of enhancement of cell proliferation both support further investigation of poly-P's role in treating AMD. And I would just like to take this moment to thank one of my most important sponsors, Bob, CBR's coffee machine. It's really played a vital role. I recommend the mochachinos. All right, thank you.
All right, hi everyone. My name is David, and I've been a co-op student in the malware lab for the past 8 months here. And I'm here to talk about the characterization of microparticles in platelet products. So over the past few years, there's been a growing interest in microparticles, specifically in their clinical role. This comes from interest in their roles in transfusion, angiogenesis, and things like cancer patients and those suffering from cardiovascular diseases, where in these patients we've seen an elevated concentration of microparticles in their blood. Now currently, flow cytometry is the method of choice for detection and characterization of microparticles, and a simpler and quicker method for detecting these microparticles doesn't really exist right now. So during my time here, I've worked with this instrument named Thrombolux, and Thrombolux uses dynamic light scattering to analyze the size distribution of particles in suspension. Now this includes things like platelets and microparticles in plasma. And in the past, we've shown that Thrombolux correlates well with flow cytometry for microparticle analysis. And with the recent interest in microparticles, we wanted to further analyze the ability of this instrument as a microparticle assay, specifically the precision of this. And we also wanted to compare the analysis of platelet samples to those of bead samples as a control. So what we did was we created platelet samples with a high, medium, and low microparticle concentration, and to accompany this, high and medium microparticle concentration bead samples. These samples were tested in parallel over 2 Thrombolux consoles over a 16-day period, and at the end of the study, the microparticle intensity and size distribution were evaluated and the precision compared between the platelet samples and the bead samples. Now we see here, this is an example, some sample results from the Thrombolux console. This is a platelet sample, a high, medium, and low platelet sample. On the x-axis here we have radius, on the y-axis the relative intensity of the particles detected. We designate everything in red here as microparticles based on size, and everything in green as platelets. And we see here that the high microparticle concentration sample, we do see a much higher, this red peak right here, and then the medium is a bit lower and the lowest. And specifically, the parameters we're looking for are the microparticle intensity, which we designate as all the area in red in these microparticle peaks, and the radius, which is the average radius in the red section, taking into account their frequency distribution. Now, if you want to see the results for the whole 16-day study, or have any other questions about my presentation, I invite you to come check my poster on poster number 10. Thank you.
Good afternoon. Sorry. My name is Eliane, and I was a summer student in the Rossi lab and also worked with the McNanny lab over at the BRC. So the project that I worked on stems from a larger study called the CHILD study, which is a Canada-wide endeavor to understand the root causes of asthma and allergic disease in children. And so because the innate immune system is largely responsible for prompting the allergic response in the body, we wondered if there's a specific innate immune cell profile in human cord blood that can predict disease development, so of asthma and allergies in children. Now, to do this, we needed to look at a wide range of immune cells. Now, typical single-cell analysis is flow cytometry, but unfortunately that doesn't give us enough depth for our study. So we used mass cytometry, which is a single-cell analysis that combines flow cytometry and mass spec. And so in mass cytometry, cells are labeled with metal-tagged antibodies, and then the presence of those metal tags is determined by their mass, by mass spec. And so we can measure a maximum of 38 parameters, so we can say like cell surface markers, and that gives us really great depth and a wide range to see what is in our sample. I showed up mostly for the preliminary part of this study. So we have the mass spectrometer. We wanted to show that it can actually do what we want it to do. So our aims were to select a panel of antibodies to detect our cell types of interest, as well as to develop standard staining and data acquisition techniques and protocols to process all of the data that we have. Now, after we run our samples that are stained with our antibody panel, we get a lot of data that can be visualized in one of these SPADE plots. So SPADE uses an algorithm that clusters cells with similar phenotypes according to the antibodies that we selected. And each kind of cell shows up as one of these nodes. Each group of cells with similar parameters. It's also organized hierarchically, so that's why it shows up as a tree. And what we were able to show is that when we look at the expression of all the different antibodies we used, this one is specifically IL-7, we were able to identify distinct group of cells, as shown by the gates. Most importantly, rare cell types like ILC2s, as you can see here, that have previously not been detected in cord blood because of the lack of technology. Now, going forward with this study means to continue running samples and then compare the data from samples of patients who eventually went on to develop asthma and allergies to those who didn't, to see if there's some sort of difference between the two, if there's some sort of innate immune profile that is predictive of this disease development. And hopefully our results, as well as all of the results from the child study, can be used to further the discovery and the development of diagnostic tests, therapies, and public health measures. I just want to give a quick thank you to everyone at the BRC, so Kelly's lab and Fabio's lab, as well as UBC Flow and AB Labs. Thank you, and the CBR.
Hello, I'm Alina. I'm from Dr. Srinathka lab, and Dr. Srinathka lab specializes on type 3 secretion system, its components, functions, behaviors. My project was about ENVH. This is a pilotin protein that binds to the ENVG as secretin and is presumed to target it to the outer membrane. During the course of those 4 months, there were a couple of findings which I would like to share with you today. The first one is that ENVH dimerizes through disulfide bonds. During isolation purification procedures, we consistently detected this second band of higher molecular weight. We assumed this to be a dimer. Upon constructing a mutation with a cysteine to alanine substitution, we did not observe this second band anymore. And this makes ENVH absolutely unique, because never before the pilotin has been reported to exist or function as a dimer. The second finding was that ENVH binds ENVG. To explore that, we employed very diverse techniques such as gel filtration column chromatography with microACTA and ITC. As for the mutant, what was very interesting was the fact that the binding to the ENVG was completely abrogated. We also did some NMR experiments, and we compared the protein alone with the second run that also contained the ENVG added. And as you see in the second picture, the peaks becomes much more clear and distinct. And now the question, whether the protein is a molten globular one, whether this is some kind of an intermediate form between the folded and unfolded states, and this is entirely possible taking into the account its disordered behavior in the absence of ENVG. So our future work might involve doing some experiments with the full-length construct, which bears a lipidation site, and doing some experiments with the binding to the real lipid. Thank you.
Hi everyone, I'm Janice from the Overall Lab. So our topic today is MMP processing regulates high mobility group box 1 activity. Sepsis is characterized by both infection and a systemic inflammatory response. It is the leading cause of mortality in Canada with a mortality rate of 30–50%. So we need a better treatment. Our current target is a nuclear protein called high mobility group box 1. HMGB1 can be secreted outside the cell and acts as a cytokine that stimulates pro-inflammatory responses and mediates downstream signaling in sepsis. The Overall Lab is interested in matrix metalloproteinase, which are a family of extracellular proteases. Using proteomic substrate screen, we found that HMGB1 is processed by MMPs. The gel shows that HMGB1 is cleaved by MMP and generates these fragments. And the cleavage sites are mapped onto the structure of HMGB1, the red one, the color. And so our question is, what effects does MMP processing have on the pro-inflammatory activities of HMGB1? HMGB1 stimulates the release of TNF-alpha and IL-1 beta from human monocytes THP1 cells. We confirmed that increasing amounts of HMGB1 leads to increasing release of these cytokines in vitro. But when we cleave HMGB1 with MMPs, the release of these cytokines decrease, and this is the gel showing the cleavage of HMGB1 with different molar ratio of MMP12. The results show that the higher the molar ratio of MMP12, the lower the production of these cytokines in THP1 cells. Next, we investigate the effects of HMGB1 on plasmin generation. Tissue plasminogen activator activates plasminogen into producing plasmin. So we confirm that HMGB1 binds to both the TPA and the plasminogen, but and enhances the rate of plasmin generation. Plasmin activity is measured by following the fluorescence of a cleaved plasmin substrate. But when we cleave the HMGB1 by various MMPs, the production of plasmin decreased. So we hypothesize that the cleavage affects HMGB1's ability to co-localize TPA and plasminogen. In conclusion, MMP processing of HMGB1 downregulates pro-inflammatory cytokine production and plasmin generation. So we should further investigate whether MMP processing of HMGB1 or the generation of HMGB1 fragments can be used to decrease the pro-inflammatory activities of HMGB1 therapeutically in sepsis. Thank you.
Hi, my name is Jody Lee and I'm from the Brahm Lab. What you see here is an enzyme CAT-K which is responsible for degrading collagen, elastin, and gelatin, and it is involved in osteoporosis. Currently, there is an inhibitor that binds at the active site that treats osteoporosis, and this drug is currently in clinical trial. However, side effects have been shown that includes skin lesions and serious respiratory infection. The goal of my project is to identify a potential compound that treats osteoporosis but binds at the active site of this enzyme, hopefully to avoid the side effects. So how do we choose these inhibitors? We screened a library of compounds using 3 docking methods and then tested the top hits. First, we did the collagenase assay, and as you can see, in the third lane, collagen is completely degraded in the presence of CAT-K and chondroitin 4 sulfate. This gel is an example. We used one of the compounds to run this assay, and you can see here there is inhibition at varying concentrations of the inhibitor. 4 of the 5 most potent inhibitors, you can see here, have a very similar 4-ring structure. Next, we did the gelatinase assay. The degradation of gelatin is solely dependent on the active site of CAT-K. Even at an inhibitor to CAT-K ratio of 20,000 to 1, you can see that there is no inhibition of the gelatin, which supports that the compounds do not bind at the active site of CAT-K. This graph here shows that there is a high percent inhibition of these compounds when they are run through elastin degradation assay. Surprisingly, when we did the binding assays, as shown here on these two graphs, there was an increase of enzyme that is bound to elastin and collagen when inhibitors were added. To sum it up, we identified 5 compounds that have an IC50 less than 15 micromolar in CAT-K collagenase assays, and these compounds do not bind at the active site. These compounds present a potential to treat osteoporosis, and further experimentation is needed to determine whether these active site 1 inhibitors have side effects in vivo. Thank you for listening.
Good afternoon, everyone. My name is Calvin, and I am part of the Prydale lab. So here we have a crystal structure of clotting factor 10A, a protein that has a role in clot formation and is essential for life. Therefore, we want to understand how this protein works. What we have found is that factor 10A has a role in clot dissolution, or what we call fibrinolysis. How it works is by cleaving factor 10A. Rivaroxaban and apixaban, which I'll simply refer to as Riva and API, are able to sit in the active site. Normally they inhibit clot formation, but what we have found is that they can also enhance clot dissolution by modifying the cleavages. So here we see the initial clot formation and then the clot dissolution. We see these samples with Riva and API have a much faster clot lysis time compared to these samples without these anticoagulants. Now, plasmin C protease have dissolved these clots. Here we're able to measure the amount of plasmin generated, and we see these samples with Riva and API produce much more plasmin. This increased plasmin generation correlates to the enhanced lysis we see on the left. Now, this is the factor 10A fragmentation, or the cleavages I mentioned in the beginning. In normal plasma, we see the formation of this band 10A3313. However, this is missing in the samples with Riva and API. Therefore, Riva and API are able to alter the cleavage and prevent the formation of 3313 while allowing factor 10A to persist. The persistent or factor 10A allows the increased plasma generation, while in normal plasma, the formation of this 10A3313 prevents it from participating in fibrinolysis. So our data shows that the fibrinolytic activity is enhanced, but we want to make sure the clot structure hasn't been affected. On the left here, we have a clot with just thrombin, and on the right is the clot with thrombin plus Riva. I was able to measure these fiber diameters, and these measurements were found to be the same, indicating the clot structure hasn't been affected by these anticoagulants. So in conclusion, we have found that Riva and API are able to enhance fibrinolysis, and this can potentially be used as a new treatment. Furthermore, or specifically, they inhibit 10A33 formation, meaning this fragment can be used as a biomarker. Lastly, we see that it enhances plasmin activity, and the clot structure hasn't been altered. If you you're interested in anything I've just talked about, you can see my poster presentation later this afternoon. Thank you.
Hey, good afternoon, everyone. My name is Mabel, and I was supervised by Dr. Agnes Lee at Vancouver General Hospital. So today I'll be presenting a study looking at catheter-related thrombosis in patients receiving high-dose chemotherapy for acute leukemia. So central venous catheters, also called central lines or CVCs, are used extensively in cancer patients and are essential for delivering chemotherapy, blood products, and for doing blood draws. Common types of CVCs include Hickman catheters. So these are inserted in the chest or in your neck and are passed underneath your skin. They are usually done in the radiology or in the OR room. A similar device called ports are also inserted in the chest, but these are implanted beneath your skin and are hidden as opposed to the catheter hanging out. Lastly, we have PICCs or peripherally inserted central catheters. These are inserted into the veins in your arms and are becoming more popular as they can be done on the bedside by the nurse. So unfortunately, there are limited studies that have focused on the incidence and management of CRTs in patients with acute leukemia. It is also unclear if the different catheter types affect the incidence of CRT. As well, there is a lack of data that exists regarding the optimal treatment of CRT, especially during periods of prolonged and severe periods of thrombocytopenia. So in order to answer some of these uncertainties, we performed a retrospective study of patients newly diagnosed with acute leukemia with a CVC inserted between January 2014 and May 2015 to determine the proportion of patients with a CRT and the incidence of CRT associated with the different types of catheters, as well as the management and outcomes of these patients. We found that in 16 of the 116 patients, the proportion of patients that had a CRT was 14%. The treatment varied significantly. So for example, the patient on the top here, he developed a clot. He was anticoagulated. His clot eventually resolved based on ultrasound findings. Patient on the bottom here, he was also anticoagulated, but then he bled, he required platelet transfusions, and eventually the line had to be removed, and he died shortly after from his underlying malignancy. So overall, our study found that the management of CRT was diverse and they frequently required platelet transfusions. The incidence of CRT was also higher in patients with PICC insertions compared to Hickman lines. And for further details and complications of the outcomes, please come to my poster after the presentations. Thank you for your time.
Hello, my name is Moritz and I spent the summer in the lab of Dr. Natalie Strunatka, where my task was to structurally characterize the alpha-2/8 polycellulose transferase of Mannheimia hemolytica serotype A2. This enzyme's product, polysialic acid, is a polymer which sometimes consists of more than 200 9-carbon backbone sugars. Interestingly, this structure does not only occur in bacteria, but as well in most higher eukaryotes, as for example mammals, where it is known to be crucial for early brain development, as it blocks neural cell adhesion molecules and thus promotes cell migration. The same feature makes it as well important for certain tumor metastasis developments. In bacteria, this polymer can be found to coat the entire surface of certain parasitic pathogens, and it is the exact same structure that is as well used by the hosts. It is non-immunogenic and can be used to evade the host immune system. Additionally, polycellulation of the surface enables those pathogens to cross the blood-brain barrier, and indeed some bacteria which possess the possibility to encapsulate, like Neisseria meningitis or E. coli, can cause severe infection to the brain. To actually characterize this protein by means of X-ray crystallography, I first had to purify the protein as far as possible, as any impurities may prevent crystal lattice formation. So I was able to optimize a purification protocol by beginning with an ion exchange chromatogram, and after a subsequent size exclusion chromatogram, I received proteins with insufficient purity for the next step. Then I had to find the environment where crystal growth could occur, and I screened several hundred conditions and finally found a crystal, but however this crystal was not of sufficient quality to be further processed for data collections. After optimizing these crystallization conditions, I obtained this beautiful, very large hexagonal shaped crystals, of which I was able to collect a structural X-ray data set with a resolution up to 2.85 angstrom, which is until now the only known X-ray crystallography dataset on any known bacterial polycellulose transferase that yet exists, which is a pretty big step in understanding the process of encapsulation in bacteria, a topic which is now trying to be understood since more than 20 years. But to finally solve the structure, then further experiments are necessary, but I'm quite confident that we can solve the structure within reasonable time. Additionally, I'm currently working on enhancing the resolution of my dataset, as this might reveal additional data on the exact mechanism of reaction of bacterial polycellular transferases. Thank you for your attention.
Hey everyone, my name is Omer. I'm a summer student in the Ellen Cote's lab, and my summer was spent researching cell-free DNA in the context of HIV infection. So first of all, cell-free DNA is extracellular DNA that is circulating in your plasma. But why is this a problem? Well, first of all, it shouldn't be there. Your DNA belongs inside your cells, and if there's a lot of cell-free DNA, it indicates that there's extra cell death going on, as you can see in the image, 2 methods of cell death. And this may be indicative of some pathologies. And surely enough, cell-free DNA has been suggested to be higher among HIV-positive individuals, and cell-free mitochondrial DNA specifically has been shown to be a predictor of mortality in intensive care units. That said, cell-free nuclear DNA has been researched a lot, but cell-free mitochondrial DNA represents a gap in knowledge, and this was part of our motivation for assaying cell-free mitochondrial DNA alongside nuclear DNA to see if they correlate to the same parameters. The objective of my project was to see how cell-free mitochondrial and nuclear DNA relate to the clinical parameters of HIV infection, including CD4 T cell count, HIV viral load, and the presence of the virus itself. So quickly going over demographics, we had 3 groups for the study: the HIV positive group on the left, that represents our study group, and we had 2 HIV negative groups on the middle and the right. HEUs are HIV negative individuals, children born to HIV positive mothers, but are not themselves HIV positive. And HUUs are healthy controls that are never exposed to the virus. So these 2 groups on the left represent our control groups. So for our methods, we isolated whole blood, and we span it, centrifuged it, and filtered the resulting plasma to get rid of any remaining cells. That way we can be sure what we're measuring is actually cell-free DNA. And we extracted this DNA and we assayed it using a multiplex quantitative PCR assay, meaning we assayed mitochondrial and nuclear DNA alongside one another in the same reaction. So this is the preliminary results that cell-free mitochondrial and nuclear DNA are higher among HIV-positive individuals, and this is what we expect, as I said before. A higher cell-free DNA may be indicative of some pathologies. This next result that we found was fairly surprising because, if you recall, I said higher cell-free DNA is indicative of something wrong, extra cell death, which indicates pathology. And you'd think intuitively that older people are in generally a poorer state of health compared to their younger counterparts. But we found a significant negative correlation according to age in both HIV-positive and HIV-negative groups, which is very surprising to us. And if you want to hear more about possible explanations for this, please come check out my poster. So I showed you relationships with age and relationships with HIV status, but how they compare when they're put together in a multivariable model. They're still both significant, meaning that age is still significant when HIV status is accounted for, and HIV status is still significant when age is accounted for, allowing us to conclude that both HIV status and age are significantly associated with cell-free DNA levels. I'd like to take this time to thank the members of the Cote Lab for their mentorship, and please come check out my poster if you have any questions.
My name is Paula, and I've been working in the J. Brooks lab this summer under Dr. Caillo. My project is called the Biological Response to Polymer Brush Coatings on Stents Under Flow Conditions. So a stent is a thin metal mesh tube that kind of acts like a scaffold to treat stenosis, which is the narrowing of an artery. Typically, there's been 2 types of stents used. The first, just being a bare metal stent, which is usually made of stainless steel. The problem with these is often reinstenosis develops. To prevent this from happening, they started coating stents with different drugs that would slowly be eluted over time. Although these were effective at the start, often thrombosis would develop later on. Both reinstenosis and thrombosis first begin from plasma proteins absorbing onto the stent surface. Once this happens, these cause resting platelets, which normally circulate in the blood, to become activated and adhere on the surface. Once this happens, and with the help from other plasma proteins, a blood clot may form. Our hypothesis is that if we apply a polymer brush coating to stents, it will act as a protective barrier to prevent the protein absorption. If we can prevent this, we can prevent the whole coagulation cascade from happening. So the aim of the project is to determine the effectiveness of the PDMA polymer brush coating on stents. To do this, we've created an in vitro blood flow loop to simulate blood flow in our body. To do this, we've created a blood flow loop as shown in the pictures below. We're using this to analyze protein absorption, platelet activation, and platelet adhesion. Protein absorption was analyzed using a fluorescent microscope. You can see in pictures A and B, which is our control sample, which is the bare metal stent. You can see how the color changed and brightened due to the fluorescently labeled protein absorbed. If you compare this to C and D, which is our polymer brush-coated sample, you can see there isn't much change in color, implying there isn't many proteins absorbed on the stent surface. Platelet activation was measured using flow cytometry and the amount of activation marker CD62P detected on the platelet surface. You can see from the graph how platelet activation varied greatly from circulation time as well as the test condition if it was resting or circulating. Platelet adhesion was measured using a scanning electron microscope. You can see the bare sample on the left, and then you can compare it to the coated sample on the right. You can see how the amount of platelets on the surface, as well as their size and shape, differed greatly. In conclusion, protein absorption, platelet activation, and platelet adhesion all decreased with a polymer brush coating.
Hi, my name is Shannon Wong. I'm a third med student from Dublin, Ireland. This summer I did a project with Dr. Heather Leach on PNH screening in MDS patients in clinical practice, the frequency and indications. So just some background. MDS is a bone marrow disorder and it's characterized by ineffective hematopoiesis and cytopenias. Most patients will develop anemia and will require transfusions. In PNH, it's a mutation in the PIG-A gene that leads to lack of GPI on the cell surface, and this allows complement-mediated lysis to occur. PNH clones are found in up to 50% of MDS patients, and this may be the reason for their transfusion dependence, and it can also help predict their response to immunosuppressive therapy. Eculizumab is the first specific treatment we have for PNH in Canada, and it helps to prevent thrombosis, reduce hemolysis and transfusion requirements, and it improves quality of life and overall survival. So with all these factors considered, we wanted to determine whether PNH as a contributor to anemia is considered in MDS patients. So what we did is we looked at 395 patients seen at St. Paul's Hospital since 1981, and we reviewed 152 of these patients seen since 2009. Only patients with a bone marrow biopsy confirmed diagnosis were included, and then we went on to look at potential indicators of hemolysis, including lactate dehydrogenase, bilirubin, etc. So this is just a summary of our results. The rectangles are showing the potential reasons for testing, and the diamonds are showing PNH testing information. So over here, there were 11 patients that had an RA or hypoplastic diagnosis, and 3 went to be tested for PNH. There were 124 patients that had an LDH and or bili measured, and 34 were increased, and out of the 34, 7 had a haptoglobin measured, and 13 had a DAP measured. Out of the 7 that had a DAP measured, 2 were decreased, and both went to be tested for PNH. Out of the 13 that had a DAP measured, 9 were negative, and 2 went to be tested for PNH. Unexplained ferritin less than 100 was in 14 of our patients, and 2 went to be tested for PNH. EPO greater than or equal to 500 was in 4 patients, and 3 went to be tested for PNH. And finally, increased transfusion requirement was in 38 patients, and 0 went to be tested for PNH. This is for a total of 11 patients, and there was 1 PNH positive. So this is just a summary of the 11 patients who underwent PNH testing. The age is again in the normal range. Gender is mostly male. MDS subtype included 1 Reb2 over here. IPSS scores were mostly low and intermediate 1, including 3 intermediate 2. Transfusion requirement was greater than or equal to 4 in a quarter of the patients. And reason for PNH testing included new MDS diagnosis, 1 iron deficient anemia, a couple hypoplastic, and 1 patient who had abdominal symptoms and had a CT scan negative for thrombosis. So to conclude, PNH was tested for infrequently in patients with MDS. Only 7% of our patients diagnosed with MDS were tested for PNH, and it was clinical rather than laboratory features that prompted PNH testing in 6 out of 11 of the patients. And although we don't really know the clinical significance of PNH clones, we do know that complement-mediated hemolysis will exacerbate anemia, and this will increase the transfusion requirements. And we do know that PNH-positive MDS patients may have a better response to immunosuppressive therapy. So now that we have an effective treatment for PNH, screening for this disorder more frequently in MDS should be considered. Thank you.
All right, good afternoon, everyone. My name is Max, and I'm a summer student at the J. Brooks Lab. So I must admit that I'm quite nervous to be standing here in front of you all today, and sometimes when I'm nervous or start using big words I don't fully understand, just to sound more hematopoietic, so please bear with me. My research has been a new project in studying synthetic polymers to neutralize the procoagulant activity of nucleic acids. To preface, thrombosis is the local clotting of blood in circulation. The SAM photo on the right shows the structure of a blood clot, and pathological clots in a vessel similar to this can obstruct blood flow. In fact, thrombosis is a major cause of mortality and morbidity worldwide. In the short time that I will be presenting today, 1 individual in the United States alone will have passed from a thrombosis-mediated symptom. Current publications have shown that elevated levels of nucleic acids in circulation and in neutrophil extracellular traps can trigger the intrinsic clotting cascade. These conditions are heavily prevalent in patients with cancer, sepsis, and infections. Hence, the objective of the project was to investigate the potential of synthetic nucleic acid binding polymers, NBPs, to safely neutralize the procoagulant activity of the nucleic acids. Our lab synthesized NBPs of different sizes with different numbers of cationic binding groups for screening. Below, you can see a schematic of our polymer containing an HPG core and positive binding groups interspaced between PEG chains. To begin my research, I first wanted to confirm the procoagulant activity of the nucleic acids and in nets. I extracted DNA containing lysate from CHO cells and stimulated netosis from neutrophils using PMA. Both the DNA lysate and nets demonstrated significant procoagulant activity in comparison to buffer controls. Subsequently, I screened various NBPs to neutralize cellular lysate and nets. Procoagulant activities were tested and neutralized in pooled plasma using a plate coagulation assay, and as well using whole blood with TEG. For the full details of my results, I encourage everyone to have a glance at my poster. Finally, to convince myself that my polymers were in fact binding and neutralizing the DNA, I activated and fixed neutrophils and stained them with a Hoche DNA dye. I then washed them again and stained it with a fluorescently labeled NBP. And under confocal microscopy, I was able to confirm the colocalization and direct binding of the NBPs to the nucleic acids within the nets. So that concludes an overview of my research project. I sincerely thank everyone from the J. Brooks Lab who have let me lead them incessantly for the past few months. Thanks as well to Libin for all of his help on the confocal images, and to Jay and Ben for their guidance. Most importantly, I would like to thank Manu for all of his experimental planning, timed so strategically so that thankfully I wouldn't need to come in on a weekend or overnight. I will come and encourage everyone to have a look at my poster and to ask questions, and thank you all for your time.
Hello, my name is Anika and I'm from the McGillivray Lab. For this summer studentship, I've been analyzing factor 7 in Quebec patients, so they have factor 7 deficiency. So to begin with, factor 7 is this shown in the pink or in the red, is a serine protease, and it's a precursor of the inactive form of factor 7 activated. And so factor 7 is activated when it's bound to tissue factor, shown in the blue, and tissue factor is exposed upon tissue damage. So factor 7 plays a critical role in the initiation of the coagulation cascade. As you can see here, that the activated form activates both factor 9 and 10, which eventually leads into the formation of a blood clot. So plasma factor 7 levels are greatly influenced by various SNPs and mutations. This can occur in anywhere of the factor 7 genes, so in any of the 9 exons and 8 introns. Therefore, a patient could be diagnosed with factor 7 deficiency. However, the symptoms could be either mild, moderate, or severe, depending on how pathogenic the mutation is. So the main goal of our project is to characterize the factor 7 gene in the Quebec patients, and from this we can find more correlations between the variations, mutations against the plasma factor 7 levels. And how we did this was in 3 basic steps. So the first one is that we amplified the factor 7 gene, and this is just through PCR, and then we verified our results with the agarose gel. Next, we purified and we sequenced the amplified fragments. And then lastly, we use a bioinformatic analysis on the genes. So we use programs, softwares, and online resources such as Mutation Surveyor, Chromis, and NCBI-BAST. And so with the results, I analyzed 15 patients. We found 16 previously identified variations and 1 novel mutation. I'll be focusing on the novel mutation for today. So the novel mutation that I happened to come across is on exon 9, and it was on base pair position 1109, and there was a guanine change to an adenine. And from here, the protein product ended up to be changed from a cysteine to a tyrosine. And so later on, we found out that this disrupted the disulfide bond, and the exon 9 happens to be the catalytic domain of factor 7. So a major mutation in this part would be catastrophic. And what was even more interesting is that we found a similar mutation, almost identical at the exact same location, except there was a cysteine change to a phenylalanine. And this mutation resulted in a huge conformational change, as well as a broken disulfide bond, and the results turned out to be quite pathogenic. So given the similarities of these 2 mutations, we suspect that this novel mutation is also pathogenic as well. However, more work needs to be done to correlate this mutation with the plasma factor 7 level of the patient. So in conclusion, once again, across 15 patients, we found 16 previously identified variation and 1 novel mutation that is suspected to be pathogenic. For our future goals, obviously we want to do more research to find more correlations between the variations and the plasma factor 7 level. Also, our current work still consists of completing the characterization of factor 7 gene in the factor 7 deficient patients of Quebec. Thank you.
Hi, my name is Kent, and I'm a co-op student in Dr. Conway's lab. My project has been investigating the structure function of CD248. So CD248 is a single-pass transmembrane glycoprotein and is expressed on the outer membrane of mesenchymal stromal cells and parasites. It is expressed during embryonic development and in adult tissues during inflammation in cancer. It's of particular interest to us because a loss of the cytoplasmic domain renders mice resistant to both tumor growth and inflammation. So my goal has been to see how this is actually happening. So in particular, I've been looking at whether or not CD248 is phosphorylated and whether or not it interacts with other proteins as sort of a mechanism of action. So for our studies, we use murine embryonic fibroblasts, and those are shown to highly express CD248. And we also have a knockout method that does not express CD248. And so I use a protein purification technique called immunoprecipitation. And when I perform this IP on both these cell types and then run them on a Western blot, you can see that a band at 150 kilodalton is visible in the lysate and the elution, and this is where we expect CD248 to be. And none of these bands are visible in the knockout. And furthermore, if you look at the unbound fraction, which is the lysate after it has been IP'd, the signal is actually significantly reduced, which is further showing that we are extracting CD248 from these cells. And then to look at whether or not the protein's interacting with these with CD248 itself, we then use a Kumasi stain on the same IPs, and this is showing us all the proteins in the gel. So here we still see the 150 kilodalton band of CD248, but in addition we see 2 more very prominent bands in the wild type that are not in the knockout, and they are likely interacting with CD248. And in the future we would like to identify these with mass spec. And then to look at phosphorylation, I use, again, IP and Western blot. Here I'm basically doing an IP with my samples and splitting them into 2 identical Western blots. And you can see on blot A we're probing it for CD248, and on blot B for phosphorylation, in this case phosphoserine. And so you can see that in all the samples except the knockout we see CD248, and particularly if you look at my controls here, on the left hand side we have the wild type in the knockout, and the CM just means complete media, and the starvation media is the SM. And that's just related to the fact that we started them overnight before these treatments on the right. And so here we see a band in the CM for wild type and in the CD248 and the phosphorylation. The phosphorylation band is slightly higher than we expect it to be, but since it's also in the IP elution, we know that it's either an interactive protein or CD248. So, and you can see that with the PDGF treatment, we also see the same band. So it's unknown yet whether or not it is CD248 or an interacting protein that's being phosphorylated, so that needs to be further proven. As well, if it is CD248 that's being phosphorylated, we want to know where it's being phosphorylated. We think it's the cytoplasmic domain, but these tests do not show that yet. Thank you.
Hi, my name's Ido, and I'm from the Kelly-McNagny group at the BRC, and we're interested in a protein called podocalyxin and its function in the kidney, specifically in a specialized cell type known as the podocyte. Just before I dive into our research, mutations in podocalyxin have been shown to bring rise to chronic and acute kidney disease, and it's also known that the majority of blood transfusion patients are actually patients with chronic kidney disease. So what we did is we knocked out podocalyxin using the Krelok deletion method. And actually, this is podocalyxin right here. It's a very negatively charged extracellular domain, and it interacts with the actin cytoskeleton. Through its intracellular domain. And basically, so yeah, we knocked it out using the Krelok method. And what we found is that these mice die between 4 and 6 weeks of age, and you can see that they die of massive fibrosis, as shown by the bright blue stain here in the Mason strike chrome stain. They also die of proteinuria, as shown by the 65 kilodalton band corresponding to albumin right here. So we said, okay, they're dying at 4 to 6 weeks of age. So we decided to look at the earlier time points and kind of elucidate why they're dying. So we picked 3 arbitrary time points: embryonic day 18.5 and postpartum days 1 and 10. And we carried out a standard histochemical analysis on them. We also did immunofluorescence and some electron microscopy. What we found is that they remain roughly the same between the wild type and knockout and the earlier time points, but as we progress to day 10, you can see some more collagen deposits down here in the Mason's trichrome and some protein deposits in the distended tubules in the periodic acid shift stain, hinting again at progressive disease state and progressive proteinuria. So then we said, okay, let's look at how quickly podocalyxin is being deleted. And we wanted to validate the fact that our progressive disease phenotype is also directly correlated to a progressive loss of podocalyxin. So what you're seeing here is a P57 stain, which is a known podocyte-specific marker, and podocalyxin, which is known to be expressed by both endothelial cells and podocytes. And what the merge indicates is that if you get a merge, then it means that podocalyxin is expressed on podocytes. But if there's a lack of a merge, it means that podocalyxin isn't expressed. So again, this confirms that the progressive disease state is actually brought upon by progressive loss in podocalyxin. And then lastly, we did electron microscopy. And this is a really busy slide, so what I want you to get from here is that here's the lumen of the endothelial cell, or the capillary, and what happens is blood flows through here and passes through the glomerular basement membrane through these things called the foot processes. And these are actually located on the podocyte. And what happens when we delete podocalyxin, if you look at the knockouts, you see that we progressively lose our foot processes until they completely melt down and basically are non-functional. And why this is because podocalyxin is expressed on the apical surface of the foot process, and it localizes actin filaments to the foot process through interaction with nerve and esrin. And because you don't have podocalyxin, the actin really just doesn't know where to go. So the question is, what's the molecular mechanism behind this thing, and can we therapeutically intervene with it to ameliorate the disease? And the answer is yes, and hopefully. So we're going to treat our knockout mice with this molecule called BizT23, and basically what it does is it promotes actin-dependent dynamin oligomerization. We're going to just see if we can ameliorate the disease state. And with that, please come see my poster. There's lots more to talk about. Thank you very much. Oh, wait. Shout out to Michael Hughes for lending me this really fancy pointer and being the greatest mentor ever, and the rest of the McNagny Lab, and Kelly and Wayne Vogel for helping with the ends.
Hi, my name is Stella, and this summer I got to work with Dr. Jackson's lab with also the help of Dr. Sun.
And my project that I helped with was a hypertension project in hemophilia patients in our province. So the reasoning for why this research was conducted was that there's been reportedly higher prevalence of hypertension in the hemophilia program compared to the general population, and that is currently unclear. So we took that as an initiative to conduct this research. Our two main objectives were to assess the potential risk factors for hypertension in our patients, as well as to evaluate the routine urinalysis and whether that gave a diagnosis of chronic kidney disease or hypertension itself. How we went about doing that was that we retrospectively analyzed 173 patients, given that they were 18 years older, having either a diagnosis of hemophilia A or B, and that they were followed by our program. We then took that data and analyzed the potential risk factors using the logistic regression analysis. What we found was that using univariate analysis, age, dyslipidemia, diabetes, and a positive hepatitis B core antibody proved to be the most statistically significant. However, a multivariate analysis, only age and dyslipidemia remain significant predictors of hypertension, as highlighted in the box above. As well, the type of hemophilia and the severity did not prove to be associated with hypertension at all. Other results that we found was that hypertension was prevalent in 34% of the population we looked at. However, less than 50% of that population are actually on antihypertensive medication, suggesting that our population is currently being undertreated. And as mentioned earlier, age and dyslipidemia are the only statistically significant factors in relation to hypertension, and there are no relation between hematuria or renal insufficiency to hypertension. Currently, there's a prospective study that's ongoing to further assess the association between renal or microbleeding and hypertension. And once the retrospective study concluded, we predict that the higher prevalence in hypertension in our hemophilia patient may not be due to the hemophilia-specific factors. Instead, it could be to that population having an increased surveillance rate than the general population. And that concludes my presentation. Thank you.
So before we start, wait, hold on, Dieter. Okay, so first, I think all the students deserve a round of applause for amazing work. And with that, all those who helped those students, and many of you are here, should be congratulated for your tremendous work as exemplified by those presentations. So congratulations. Now, I just asked, this is a real tough one, so I'm sure there's going to be a lot of ties. So you two rows of students in your scoring scheme, I'd ask that you indicate your total scores on the right-hand column, and then just put a 1, 2, and 3. I'm sure one of those 1, 2s, and 3s will be yourself. And then pass those papers to your left, okay? So please do that. And while they do that, first, before we move on, I do want to take the opportunity to thank many, many people. In addition to our sponsors who are posted here, all those volunteers who have helped in set up and hopefully take down of this event. It's very much appreciated. Hannah, is Hannah here? Hannah's there. All right. So Hannah has really, this is her first time doing this, and she's really done it single-handedly. Anna is away this week, as you know, but Hannah's sort of taken responsibility, and I've sort of left her. And as you can see, she's done an amazing job, and you can see outside she continues to do a great job. So please thank her. I'd also just like to thank Ed Prysdale. Ed, are you there? Are you there? Okay, the associate director of the CBR, who's been instrumental in helping me through this and organizing. These students have had an amazing summer. They continue to have an amazing summer. I've heard lots of reports. They've drank more beer probably than they should have. They've gone on work canoes. They've done all kinds of social events, but they've also had sort of career advice. I'm not sure if it's advice really, but career input as to what not or or what to do or what not to do, and I'm sure they'll all be great scientists, needless to say. But opening up other opportunities, they've gone on tours of various facilities that I haven't seen, but I know that they've been terrific. So, and a lot of that is the hard work, again, of Anna and Hannah. It's really easy to have two people like that in my office. So now I'd like to move on to the next part. So Neil Mackenzie, and I'm going to ask Dieter to introduce and tell us a little bit about Neil.
So I want to also congratulate all speakers for the great talks, very enthusiastic, and some of them are very entertaining. So you will get, I think, a prize after that in a few hours when the poster session is over. But we want to honor the first time also the other part of you guys who are mentoring you. So the mentors were essentially our graduate students and postdocs. So, and we want to really the first time give them an award. And we generated this award in the name of Neil Mackenzie. So who is Neil Mackenzie? Neil Mackenzie was a postdoc of mine, joining in summer of 2013, coming from Scotland, a real Scottish guy, as you can see probably, red hair, beard. So he was a great postdoc in my lab. And he started a project, actually we got a CIHR grant, a part of his efforts. We wrote the grant together when he was still in Scotland. And joined my lab, has done great work. He was a really good mentor to students, was enthusiastic all around, and he loved nature. He was outside, he was a mountaineer. And unfortunately, I guess a lot of you know, he passed away beginning of the year in a tragic mountain accident together with two other friends of him. And so we generated this mentorship award in his name, and I think it's full in his honor, and it fits perfectly to his character, the way he was. And so we had 30 nominations. I think we had around 30 presentations, or a little bit less, and 30 nominations. And you had to write actually 500 words of praising your mentor. And so we had to figure out now who is the winner. We have unfortunately only one prize. I don't know, I was not among them, I don't have the name, so I give it back to Ed, and he will announce the winner. So one single winner this year.
You can announce the winner. But just to point out that there were many nominees, and the writing, I had the opportunity to read. I did not have any input in terms of the award. The awards, the awards were judged from outside of the CBR, so nobody really, none of those had any sort of influence, just in case you were wondering, and it was really a difficult decision, but nonetheless it was unanimous indeed. So here are the nominees. All the nominees will receive an award, and those people should come to Hannah, to the administration office, CBR administration office, sometime in the next week to pick up that award. But the winner of the award was selected to be Steve Herr. Come on, do you want to take pictures?
3, 2, 1.
Thank you. So this is from Mac. We thought that this was appropriate, that you should have something that's climbing related. There is a headlamp here in case you get lost on your way to Mac. Okay? Thank you. The next part of the program will be chaired by our own Ross McGillivray. In case you don't know him, he was the founding director of the CBR, but everybody knows him. Can't keep your
Time's up. Okay, well, thank you, Ed. It's a real pleasure for me today to introduce Bill Milsom, who's been a wonderful colleague here at UBC, and I think we've overlapped for 35 years, which is a little bit older than most people have been in the world in the audience. Bill is originally from Toronto, but was the son of a military family and travelled across Canada as a kid until settling on Edmonton for his undergraduate studies at the University of Alberta. And then he did a master's at the University of Washington in Seattle before coming back to Canada and doing a PhD with the late David Jones, who many of us really knew and liked a huge amount. Just a great guy. Bill then stayed on, became a faculty member in zoology at UBC, and had the pleasure of being head of department for 10 years, 2002 to 2012, and then, 2013, followed by being the director of the UBC Fisheries Institute. Bill has been an incredible teacher of biology on campus, teaching everything from introductory biology to all sorts of evolutionary and environmental physiology. He's authored over 200 papers and combines that with being a co-author on 2 textbooks. He is also on the editorial board of 7 journals and currently has many interests, all surrounding evolutionary and environmental adaptation of animals to certain conditions. And I had the pleasure of having a beer with Bill about a month ago on the occasion of his retirement, but I've since realized he's not really retired, he's just not being paid anymore by UBC. Because when he kindly accepted our invitation to be the keynote, he was teaching in Brazil. And then when I asked him for his brief bio, he was just returning from Peru, where he'd been continuing his research. So Bill is a wonderful colleague, a wonderful academic, both in teaching and research. And today he's going to tell us words of wisdom surrounding geese that fly high. It's in their blood. So please join me in welcoming Bill.
Well, it's a pleasure to be here. As Ross said, now that I'm retired, I actually have the privilege of saying no to anything anybody asks me, but as this indicates, I'm not very good at saying no. So, my lab is a little bit schizophrenic. A big part of my lab is interested in fundamental mechanisms in respiratory rhythm generation, respiratory pattern formation. The other half of my lab is interested in how those fundamental mechanisms can be adapted to allow animals to live in extreme environments, places that are not hospitable to most. And amongst those, the story I'm going to tell you today is one that's almost complete, but still a little bit ongoing, and it's about a particular bird, and that's the bar-headed goose. Now, the bar-headed goose, what makes it outstanding is this biannual migration between its wintering grounds, where it feeds and fattens in India, to its breeding grounds up on the Tibetan and Mongolian plateaus. And to get back and forth twice a year, it actually has to fly over the Himalaya, over the highest mountain range on Earth. Now, of course, going to altitude is challenging at the best of times, but I want you to remember that flight is probably the single most expensive form of locomotion. And at altitude, it's even more expensive. The air is thin, which reduces lift. It does reduce drag a little bit, but more so it reduces lift. It's extremely cold. The air is dry. And from our perspective, the most important thing is that oxygen is very limiting. Now, to put it in perspective, it wasn't until the early 1980s that humans made it to the summit of Mount Everest without supplemental oxygen. Even today, most climbers going above 7,000 meters use supplemental oxygen, which they carry with them to enable them to climb at those altitudes. For those that have made it to the summit of Everest without supplemental oxygen, if you read their accounts, as they near the summit, it's one step and rest, one step and rest. Yet these birds are able to fly at those altitudes, and as I'll show you shortly, they do it with no acclimatization. So the big question is, how do they match the demand for oxygen with the very limited supply that they face when they're at that high? Well, when we started this, the first question we had to ask was, how high do they routinely fly? Because that would give us an idea of what the O2 supply was. At that point, most of the evidence was anecdotal. There were a couple of anecdotal studies that said that they actually flew over the summit of Everest, but there was no hard data. And so the very first thing we did then was we started doing GPS studies and field studies in Mongolia. We go to Mongolia because once the birds breed, they molt. When they molt, they can't fly. When they can't fly, we can actually catch them. There we started then doing surgery in the field to implant physiological data loggers that allow us to measure a variety of different variables, but we also mounted GPS units on them. We learned the hard way. The one that's actually shown in the bottom of this figure is a solar panel GPS. I can tell you that they did not work because the birds simply preen their feathers, covering over the top and blocking out the sun. So we actually had to go with battery-operated units to finally get data. What that data showed us gave us an idea of the tracks where they were flying. And although you can't see it all that well here, if you take a look, in gray is the Chinese plateau. To the south of it, in dark brown, are the actual Himalayas, and the birds are actually flying through Bhutan. And the amazing thing when we sat back and took a look at all of this, and you can see it on this slide, is the Indian subcontinent is actually fairly flat. There aren't too many points above sea level until you hit the base of the Himalaya, at which point it rises dramatically and rapidly. So we found then that the birds, a number of things. The first one is that they routinely cross, but they're not dumb. They go through the passes. So that typically they're going between 5,000 and 6,000 meters, which is where most of the passes are. And only on rare occasion did we have recordings of birds flying higher. The highest that we got was 7,300 meters. So this gives us an idea of supply. Just to put this in perspective, at 5,000 to 6,000 meters, the air is roughly half of sea level oxygen, 10% rather than 21%. The summit of Mount Everest at 8,800 meters, it'd be down to about a third, roughly 7% inspired oxygen. So we know these birds then, the amount of oxygen available is going to be in the range of 7 to 10%. So then the next question was, how metabolically challenging is it? How much oxygen do they need? And of course, one of the things that people had always speculated was the extent to which they could use the updrafts, because you watch paragliders and hang gliders, they'll always use during the day the thermals rising up the face of the mountains to give them lift. Well, if you take a look at a bar-headed goose and compare it with an albatross, you can quite clearly see that a goose is not built to glide. And in fact, the comparison I use is with a glider, and on the right is the Drummond goose, which is being put into wide-spread use this summer for fighting forest fires. We did find, in doing an analysis now of over 10 different species, that relative to their body mass, the barheads do have a slightly larger wingspan than you would predict for their size, and as a result, the wing loading is lower than you would predict for a bird of that size, but they're still not gliders. And then the data that come back off the GPS units, which tend to support this show, if you take a look here at the elevation profile, and these are the birds leaving India flying north, and you can see that they hit the face of the Himalaya and they go up rapidly. Each trace is a different bird. And the time of day profiles show the wind direction and the time of day they're flying. And notice that they tend to fly during the night, when in fact there's a small downdraft. And for the most part, they avoid flying during the daytime when the updrafts are there. We think that there might be too much turbulence, and they prefer the cooler at night, which will also lower barometric or raise barometric pressure and give them a little bit more oxygen. But also it's calm, and that seems to be the preferred time for them to cross. Also notice that they're leaving from sea level and they're making it to 5,000 to 6,000 meters in under 8 hours. There is no acclimatization. There's no preparing to get to elevation. They just leave and they do it. Now, the other thing that we found, which was interesting, is you might predict that, and this is the profile for a single bird. I'll see if I can do this with the cursor here. That's showing up, yeah. Is that rather than when they begin to peak, then staying on the dotted blue line and staying high until they get over the highest of the mountains, they actually follow the terrain. So they're going up and down and up and down. And you can see off of this then that rather than ascending what would be a gradual from 3,000 meters to about 4,500 meters, they actually went up and down 6,300 meters of ascent and descending almost 5,000 in that roller coaster. And just based on how hard it is to work in the thin air, it's actually, with modeling based on drag and lift, it's actually cheaper in terms of oxygen consumption to follow the ground, even though it means going up and down so much more, than to stay high and fly at those very high altitudes over that range. Well, so it's indicative that it's a lot of work, but how much? How much oxygen do they need? We find two things from our GPS units and our data loggers, and that is, you can see if we go from red to green to blue, we're going from our average values at 4,800 meters, or sorry, red is about 2,300, green is up to 5,000, and blue is above 5,000 meters, that their heart rates and their wing beat frequencies are going up progressively. So that they're having to flap harder as the air gets thinner, the heart is having to work harder as the air gets thinner, and they have to flap more. That might not be too surprising. One of the most difficult things to do in the field is to measure oxygen consumption of wild animals. The best method right now uses doubly labeled water, but it gives you a measure from when you release the bird to when you catch it again, which isn't going to tell us much about what's happening during that flight. So there is, though, depending, as long as other variables stay consistent, a reasonable correlation between heart rate and metabolism. So one of the things we then set out to do was to see if we could see what that correlation was, and then use heart rate to give us an estimate of metabolic rate. And to do that, I had a postdoc, Jessica Meyer, and a couple of undergraduate students, Julia York and Michelle Reichert, and they raised bar-headed geese so that they're imprinted on them from the moment they hatch. So this is their surrogate mom, whom they will follow anywhere. So that as they begin to fly, they will follow her on her bike, and then on a motorcycle, and then ultimately into the wind tunnel. And try to bring this up now. There we go. And this is a slow motion film footage of the bird in the wind tunnel. As the wings come down, you'll see there's a little backpack on the bird. They have an arterial cannula, a venous cannula. We measure body temperature. The mask allows us to draw air to measure metabolic rate, and there's a second line there that allows us to deliver oxygen so we can progressively drop the O2 to simulate and have the birds fly right up to 7% oxygen, equivalent to almost 9,000 meters in the summit of Mount Everest. So the net result of the wind tunnel studies, we're able to show that indeed it's not tight, but there is a correlation between heart rate and metabolic rate, from rest to walking to treadmill running to flying in the wind tunnel. And from the GPS data, we can see there's a good correlation between wing beat frequency and heart rate. And so there's nice, reasonable correlation between all of then wing beat frequency, heart rate, metabolic rate. And based on that then, we can come back and we can look at our heart rate, and we see that on average over that whole migration, heart rates for these birds were reasonably low. High for us, 300, 350, but at times they're going up over 450 to 500 beats a minute. And at those levels, our estimates then are that their metabolic rates are 16 to 20 times resting values. So they're elevating their consumption almost 20 times in air that has half to 1/3 of the amount of oxygen. So then the big question comes, how do they do it? And I'll go through this fairly quickly, but to do it, they have to move oxygen from air to mitochondria through the oxygen transport cascade. 5 steps. Ventilation, they have to get it into the lungs. Pulmonary perfusion, they have to move it across the lung into the blood. Perfusion to deliver to the tissues. Tissue diffusion to get it into the mitochondria, and then of course cellular metabolism and mitochondrial oxygen consumption. Now I can tell you right from the start that birds do better than mammals at every step in that cascade. And I won't go through the details, you'll have to trust me on this, but the bird lung is designed very, very differently so that it is a much better gas exchange surface. We see in general, birds have, relative to body weight, much larger hearts, a higher capillarity in the muscle, a whole slew of adaptations that all birds have that are associated with flight. The question then becomes, since not all birds can fly at altitude, what then do the bar-headed geese have on top of this that the other birds don't? And very quickly, we see adaptations at every level in the cascade. So in terms of ventilation, if we make them hypoxic and drop arterial O2, you'll see that in yellow, the bar-headed geese increase their breathing earlier and are more sensitive and increase it and have an enhanced hypoxic ventilatory response. At the level, whoops. Oh, no, don't sand, just give it back. Let's try that again. Okay. Can I go quickly? Make sure it doesn't crash on us again. Okay, an enhanced response. At the level of pulmonary diffusion, and here are data just for 2 birds of exactly the same weight, I think they differed by 50 grams, that we ran CT scans on. Barhead on the left, a barnacle goose, a low altitude migrator that flies from Svalbard down to Scotland and back every year. Good migrator, but you can just see by looking, the lungs are much larger in the barhead, much more surface area for gas exchange. The air sacs are much larger. The trachea is wider, less flow resistance for the movement of air, and there's more air in the long bones, so that for the same volume of bird, the bar-headed geese are much lighter. More air within the bones themselves. At the level of the exchange surface, this is work that's still in progress that we hope to finish by the end of the year. It looks like there may be a small difference in the barrier thickness between air and the erythrocytes. Should point out that bird lungs are very, very rigid. It's the air sacs that expand, the lung does not. The net result is the barrier thicknesses in birds are down to about 1/3 to 1/5 the level of a mammal, so that it's a much shorter distance across in all birds. And it looks like the barhead, it might even be a little bit thinner. I'm going to jump over perfusion and go to tissue diffusion for a moment, just to point out very quickly that if we take a look at muscle from the surface, intermediate and deep, with the surface muscles being the ones that power the downstroke and are most associated with generating lift, that we see that there are more type 2A fibers, so more oxidative fibers. We see that the mitochondrial volume density is higher, so that they have a higher aerobic capacity. We see that for mitochondrial density, they have more capillaries, so their capillaries are closer to the individual muscle fibers. And finally, you see on the right that most of the mitochondria sit right under the sarcolemma rather than deep within the muscle, so that the diffusion distances for oxygen are reduced. So again, we see a whole slew of adaptations at the level of tissue perfusion. At the level of cellular metabolism, quite simply, mitochondria are mitochondria for the most part, so that we find that the oxidative capacity is not enhanced. If we look at a whole variety of different enzymes for aerobic metabolism. There are very little differences between the high altitude and low altitude species. And we also find that there are no differences in the O2 kinetics within the mitochondria themselves, without going into detail. The only thing we found that there was any difference in was in the cytochrome oxidase enzymes within the mitochondria, but very, very minor. So now let me come back to where the title of this talk comes from. The middle is perfusion. And for perfusion, the Fick equation, oxygen consumption is going to be equal to the amount of blood being pumped, heart rate and stroke volume, the carrying capacity of the blood, beta, and how much is extracted as the blood goes through the tissues, the difference between arterial and venous. So there are three things you can do to increase O2 delivery. Pump more blood, carry more in every mil, or extract more out of each mil as it goes through the tissues. You can rewrite those equations. I'll just jump to the bottom one. Because beta is going to reflect hemoglobin concentration, hematocrit, mean corpuscular hemoglobin concentration, and all of that's going to determine the relationship between the content within the blood and the partial pressure. I can say that the hematocrits and the hemoglobin concentrations are no different between the high altitude species and the low altitude. So that narrows things down then to either the changes in the cardiac output or the changes in the carrying capacity of the blood itself. Okay. Well, if we take a look, and quite simply, a complex diagram of cardiac output, which is stroke volume times the isopleths, the frequencies, you'll see there are no difference between species. So their ability to increase cardiac output is the same, and really where we see the difference is in the oxygen equilibrium curve. And if I just put the lines up quickly, the purple is the low altitude bird, and you can see that it begins to desaturate much more quickly. The other lines, the green, are bar-headed geese raised here in Vancouver at sea level, and the blue one are wild bar-headed geese captured in Chinese Tibet up at Lake Qinghai. And you can see a dramatic difference in the carrying capacity of the blood between the barheads and the barnacles, and between the wild barheads and the captive ones raised at sea level. So there is some phenotypic plasticity as a function of where the birds are raised. The biggest difference we know is due to a single amino acid substitution in the beta chain between the barheads and, in this case, gray lag goose. And that one single amino acid substitution leads to that dramatic increase in the ability of the hemoglobin to bind oxygen. The other thing we know is that the barhead blood is much more temperature sensitive. pH sensitivity is no different. Bohr effect is the same, but it's more temperature sensitive than the blood of other geese. And our hunch is, and I'll take a moment to go through this, if you follow along the red line, which is the oxygen equilibrium curve, birds are typically 41 to 42 degrees. They're much warmer than we are. And 4% CO2 is normal for bird blood. If you follow along that line and you look at the heavy red arrow on the left, this would show you the amount of oxygen that would be unloaded between arterial and venous blood for blood going through the body of the animal. Our hunch, though, is that flying at altitude, where the temperatures are minus 20 to minus 30 degrees at that altitude, that the lungs are actually going to cool. And they're very alkalotic because they're breathing so hard. Whereas as that blood gets to the muscle, which is generating all the heat, the temperature will rise and it will become acidotic due to the production of CO2. And that difference between cold alkalotic blood within the lung and warm acidotic blood within the tissue can more than double the amount of oxygen that's released as the blood goes through. Does it do that? Well, we've gone back to Mongolia. There's our field site down on the right. There's the local nomads on the left who provide us with food. And we've done surgery in the field, and what we've implanted into these birds are two loggers. The data logger measures pressure to give us altitude. It also measures 3D accelerometry so we can get wing beat frequency. We've measured the temperature within the flight muscle, the temperature within the lung itself, and on the collar, we've also got the temperature of the ambient air. We released 40 of those a year ago. We've gone back, and if you look at this picture, somewhere out there you can see the ones with the green neck collars. We actually managed out of 16,000 birds to recover 30 neck collars, which was a massive 3-week effort. And those collars are all now sitting with my colleague in Tasmania, waiting for the data to be downloaded. And this is the last piece of the study that I can't give you the answer for until next year. But in summary, we see adaptations at every stage within that cascade that enhance the ability of the barheads over that of other birds to do this remarkable fleet. Now, the one question that we'll probably never answer is what can they do? Here's a quote from Lawrence Swan, who was one of the people who first anecdotally reported the barheads flying over the summit of Mount Everest. We know they can go through the passes. What we don't know is whether they actually can or cannot go over the top. It would be extremely lucky to have a unit on one of the birds who did that. Our hunch is, though, that the weather there is inclement as often as it is around here, that if they hit the mountains in a whiteout, they may go that high. Maybe one day we'll find them. So at that point, I just want to acknowledge all the people that have been involved in these studies that have been ongoing for the last 5 years, and thank you for your attention.
Thank you, Bill. Would you mention a couple of questions if people have them?
Certainly.
Any questions for Bill? Cedric?
Famous physical country skiing with aeronautical answers. You get a hemoglobin out all the way through. Do you know it?
In the hemoglobin?
Yes.
So the hemoglobins, we are finding that the hemoglobins in the birds are, I was looking at the slides earlier, we can get some up into the 230 range. They can be high in the high altitude birds, but we find that they can be equally high in some of the low altitude migratory birds as well.
Had that mutation. You mentioned that mutation.
So the mutation in the hemoglobin then means, and the downside, I should point this out particularly to the students, when anybody talks about adaptation, what's the trade-off? So with a hemoglobin that binds a lot of oxygen at low, it's not going to give it up easily. So how does it get off in the tissue? Because when it does, the perfusion pressure is going to be very, very low between blood and mitochondria. There's always a trade-off, and we assume that the high capillarity and the shorter distance between mitochondria and capillarity balance that out. But for all the other adaptations, anything that's going to make it easier for a barhead, why doesn't every bird do it? What's the trade-off? Because there are always going to be trade-offs.
Peter?
Do you know what is the highest altitude ever recorded on bird? Somewhere around, was it say claims they are 10,000 meters with the airplane?
Yeah, so they have found some of the bustards and vultures which do soar and glide up over 10,000 meters. And they only know that because they ran into planes and got sucked into the engine. So we know that birds can go that high, but almost all of those records are non-flapping. So maybe a bird usually caught in a storm with a lot of uplift.
It's a selective disadvantage too.
Don? That graph you had of the distribution of heart rates, there were a lot of birds down at the low end. Are they super birds or?
So what those curves are then is they're the Bell distribution of the heart rates from, and I think in that case, 11 birds over the entire migration. And so off the GPS unit, we've simply taken, okay, anytime they're over this elevation, what are the heart rates? And so it's just giving you a mean distribution.
Yeah. So is there a future for xenografts, like lungs from birds and humans for disease?
Only if you get air sacs to go with them, since they won't expand.
You have to be excited too.
Just a little bit. Okay. Any other questions?
I'm wondering if you can draw any parallels with the Tony Farrell's work and the adaptiveness of salmon to different temperatures owing to different streams with different temperatures.
Well, one of the things that is of interest, of course, with climate change is just what will happen, because we assume that cold air is a benefit for a variety of reasons, both for loading oxygen into the hemoglobin if the lungs are on the cool side, and also because at the colder temperatures do influence barometric pressure and also make it a little bit easier for them to fly. So if we do get global warming, what will the consequences be for that migration? At this point, can't predict, but with a little bit of modeling, perhaps.
Okay, well, on behalf of the CBR, thank you so much, Bill. And,
And time for a beer. Oh.
Thank you.
Thanks so much. All right, so that really wraps up this session. For two things. One, students, please stay behind. Bill, stay behind for just a picture of the group of all the summer students. Then there's beer and wine waiting for you. So as you walk out, just make a left into the atrium and it'll be sitting waiting for you. Posters are out there. Between 4 and 5, I ask the students to be around your own poster so you can be judged and you can describe what's going on, and after that you're free. There'll be a barbecue starting, food will be served around 5:15, 5:30, and the awards will be given at that time for the poster and the best oral presentation. All right, so thank you everybody.